These videos are offered on a pay-what-you-like basis. You can pay for the use of the videos by making a monthly pledge at my Patreon page. Or you can make a one-time payment by using the PayPal donate button on my website. Thank you. Did you have a chance to memorize the amino acids?
No, I haven't had a chance to
Okay,
memorize.
that's a good thing to do. Because remember, next week you'll be on to another chapter and you'll have a bunch of new things to memorize. And then the week after that, you'll be on to some more chapters and you'll have a bunch of new things to memorize. There's a lot of memorization in biochemistry. So there's going to be a big load of things to memorize each week. There's probably more memorization in biochemistry than in organic chemistry. In organic chemistry, we kind of tried to focus a little bit more on understanding, so we didn't have to memorize as much. Well, obviously in biochemistry, we want to understand as much as we can, but there's a lot of memorization. There's a lot of stuff that just has to be memorized in biochemistry, and you can't kind of let that pile up because then there'll be too much. No time like the present, no time like this weekend. That's a really big job to memorize all 20 amino acids. That's an important thing to start working on. Okay, well, let's do this. First of all, there are four main types of large biological molecules. Your biochemistry course is going to focus mainly on these four types of large biological molecules. Do you remember from class or from your biology class, what are the four types of large biological molecules?
Is it starches? Starches.
Anything else?
Polysaccharides, like sugars.
All right. Anything else?
Amino acids.
Okay, so we're not quite getting the correct terms here. So one term is carbohydrates. You mentioned polysaccharides, that's a type of carbohydrate. And you mentioned starch, that's a type of carbohydrate. Then there's proteins, lipids, and nucleic acids. So these are the four main types of large biological molecules: carbohydrates, proteins, lipids, and nucleic acids. Do you know what the two main types of nucleic acids are?
I just wrote them down today. I don't know. Yeah.
All right, so these are molecules you should have heard about in some of your other courses as well. What do you think the Na stands for in DNA?
Nucleic acid.
That's right, and the same in RNA. So nucleic acids are these very important molecules, DNA and RNA. So your biochemistry course is basically going to be learning about each of these four types. We're going to be learning about them one at a time. What is your class focusing on now? Proteins. So we'll focus on that. First of all, tell me when you have these four types of biological molecules memorized.
Okay.
Okay, well, without looking at your notes, what are the four types of large biological molecules?
First is carbohydrates.
Good.
Then lipids.
Mm-hmm.
Then proteins.
Mm-hmm.
And then nucleic acids.
Good. And what are the two main types of nucleic acid?
DNA and RNA.
Good. And you don't have to memorize them in any particular order. There's no meaning to the numbers here. It's just that there's four of these. Okay? So now let's focus on proteins. Now, proteins are a polymer. Do you know what a polymer is? A polymer is something that consists of many repeating subunits. Kind of like a train is made up of many cars. If you take a bunch of cars and you link them all up, you get a train. And you can add more cars to get a longer train, or you can split the cars in two to get 2 trains. Many large biological molecules are polymers. What do we call a protein polymer? Protein polymers are called polypeptides. Well, actually, you could just call it protein. Or you could call it polypeptide. Or sometimes they're just called peptides. So these all refer to a protein polymer.
Okay.
Now, usually the word protein is used for the longest polymers, and maybe polypeptide might be used for an intermediate-sized polymer, and maybe the word peptide would be used for a fairly short polymer. So sometimes this is for longer and these are for shorter, but sometimes these are used interchangeably. People are not that consistent. All of these refer to proteins. Now, what is a protein monomer? Remember, the monomer is the repeating subunit. Do you remember what's the name of protein monomers?
Amino acids.
That is the right answer, yes. So is an amino acid a protein?
No.
No, it's not. Remember a few minutes ago I was asking you to name the large biological molecules, and at first one of the things you mentioned was amino acids. Well, that was on the right track, but amino acids are not large. Amino acids are a building block for proteins. So oftentimes students confuse proteins and amino acids. So what's the relationship between the word protein and the word amino acid?
A bunch of amino acids make up what a protein is.
That's right. So this could be an amino acid, 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 and the whole thing together
Is a protein.
is a protein or a polypeptide. Okay, so it's important to get these terms straight. How many amino acids are there?
20.
Yeah, there's 20 common amino acids. So that's good news for life because that gives us lots of variety for making proteins, but that's bad news for biochemistry students because it means 20 things to memorize. There are 20 amino acids. Now, all amino acids have very similar structures. They all look like this. This is the structure of an amino acid. This has to be memorized.
And the R group dictates what kind of amino acid it is.
That's a good point. Excellent. Do you know what the name of the R groups are in general then?
I don't.
So these are what are called the side chains. But your point was a good one. So all amino acids, this portion is the same for all amino acids. So this is what I call the main chain. I think sometimes this is called the backbone. Called the main chain or the backbone. These are the same for all amino acids, and the side chains are different for all amino acids. That's why I wrote it as an R, because I don't know what this is if I don't tell you what the amino acid is. So let's see, what are the functional groups in the main chain? What type of functional group do we have here in the main chain?
Carboxylic acid.
That's right. So all amino acids have a carboxylic acid group. And what type of functional group is this?
An amine.
Yeah. why are amino acids called amino acids? Why is that a logical name for them?
Because they have an amine and an acid.
That's right. So it's a very logical name. I guess it would be more logical if they were called amino carboxylic acids. That would be a little bit too much to say every time, so the scientists shortened it to amino acid. But this just refers to the amino group and the acid. Notice it's not the amino group. Well, anyway. Okay. And then we also have the side chain. We need to give names to all these carbons. So this carbon here is the carboxy carbon. Or maybe we could also call it the carbonyl carbon, if you remember the idea of a carbonyl from organic chemistry. That's the carbonyl carbon. So we need a name for this. Now, we also named these in organic chemistry. Do you remember, what did we call the carbon connected to the carbonyl carbon?
Alpha carbon.
So let's keep using that name. This is what your textbook calls it. This is a very important name. This is the alpha carbon. So the basic structure of the amino acid main chain is nitrogen, alpha carbon, carboxy carbon. That has to be memorized. Nitrogen, alpha carbon, carboxy carbon. Nitrogen, alpha carbon, carboxy carbon.
Hold on. Okay.
Okay. So I'll write down a couple of examples. So this is valine. Remember, you also have to memorize the 3-letter abbreviations and probably the one. Did you ask your instructor if they want you to know the 1-letter abbreviations?
They don't.
Oh, really? Okay, that's unusual. Most classes you need to know that. Okay, but you do need to know the 3-letter?
Yes.
Okay, so that has to be memorized as well.
Yeah.
Pardon me?
Nothing. Sorry. We also, just as a quick side note, we did the pKa's of functional groups, and she says that most biochem classes require you to memorize them. And she says just know the general ballpark of them, and I would rather have, memorize them instead of just knowing the general ballpark of what they are.
Okay, so we'll talk about that. All right, so I just wanted to write down 3 examples here of amino acids. These are all examples that you're going to want to memorize. What's the name of this carbon?
Carbonyl carbon?
Yeah, or the carboxy carbon. And what's the name of this one?
Alpha carbon.
This carbon you could call the beta carbon. This could be called the beta carbon. Is this part of the main chain or the side chain?
That is the side chain.
How about this carbon?
That is part of the main chain.
How about this carbon?
Main chain.
How about this one?
Side chain.
How about this one?
Side chain.
How about this one?
Main chain.
So it's very important to see where the side chain starts and the main chain ends. The alpha carbon is part of the main chain. The beta carbon is the first carbon in the side chain. Okay, Now, here, what type of functional group is this here on the right?
Carboxylic acid.
What are the two forms of a carboxylic acid? What's the other form of this carboxylic acid?
The deprotonated form, the conjugate base.
How would I write that?
With the negative oxygen.
Okay. Now, it turns out that we were talking about memorizing the pKa's. So the pKa of this carboxylic acid here, the pKa is approximately 2. What's physiological pH?
7.
So at physiological pH, what form will this take in the amino acid? What will be the major form?
The major form would be the deprotonated form.
And what's your logic there?
Because the molecule is more acidic. No, so it would be the protonated form because the molecule is more acidic. Wait, when it's done donating the proton, it's going to be the deprotonated form because the molecule is more acidic, and when it's done donating, it's going to have a negative charge.
Okay. So your first answer was correct. Then you had some second thoughts, but you went back to the correct answer. So the molecule here is much more acidic than the solution. So the molecule is going to give its protons to the solution and be left with few protons. So what we just saw here is that in an amino acid, amino acids generally take a you don't actually usually write the carboxylic acid as an acid. You usually write it like this, with a negative charge. Now we need to analyze the left-hand side. What's the other form of this left-hand side?
The NH plus. NH3 plus.
We have to get the right number of protons. And the pKa of this amine group is approximately 9. So we were just talking about ballpark numbers. So here's 2 ballpark numbers to memorize. I think this is what your instructor meant. The ballpark for carboxylic acid is around 2, and the ballpark for amines is around 9. So I think these are the types of ballpark numbers that you need to have memorized. So at physiological pH, what's going to be the major form of the amine side?
At physiological pH, that's pK of 9, right?
Yes.
It's going to be the positive charge.
Protonated or deprotonated?
Protonated version.
What was your logic?
The solution is more acidic, so what it's going to do is the solution is going to donate protons to the molecule, and when the solution is finished donating protons to the molecule, that's what it's going to look like.
Okay, that's right. So at physiological pH, what's the charge on an amino acid? The total charge.
Neutral. Because
the positive balances the negative. The positive balances the negative, and overall it would be neutral. So when you're memorizing the amino acids, they're actually, you'll see in the textbook, they're usually actually written like this, rather than in the neutral forms, because we're studying biochemistry, so we want to know what these look like in the body. So here's some of those, so here's 2 numbers to memorize, these ballpark numbers. The carboxylic acid side is around 2, and the amine side is around 9. You could see a test question where they asked you, in fact, you're very likely to see a test question where they ask you what would the amino acid look like at a different pH. In fact, let's do that. So here we had a physiological pH 7.4. How about let's do a pH of 11. And let's draw what would valine look like. What would valine look like at a pH of 11? Take your time and try to work that out.
So instead of 7.4, it's 11.
The pH is now 11, yeah.
The it would have a deprotonated carboxylic acid. Well, it would have a negative charge on the oxygen. Bonds.
You cut out for about 10 seconds there. So the last thing I heard was that there would be a negative charge on the oxygen. Is that right?
Yeah.
And then what was your next analysis?
Since the solution is less acidic than the amine group on the molecule is more acidic, it's going to be donating protons to this. The molecule is going to be donating protons from the amine group to the solution. It will have a neutral form.
On the right or the left?
Left.
Protonated or deprotonated on the left?
Deprotonated.
Like this?
No, NH minus.
Like this?
Yes.
Okay. So there's a little confusion there. So what does the amine look like when it's neutral?
NH2.
Now, each of the groups we're going to be studying here only has two forms. What's the other form of the amine?
NH3 plus.
We're not going to study the NH minus form. I think we talked about that earlier. Is this neutral form, are we treating the neutral form as an acid or a base?
The base.
So when it picks up a proton, it's going to look like this, NH3 plus. We're never going to get to the NH minus. There is an NH minus form, but that's not going to come up in your course. Since we're only treating this as a base, that form is not going to come up. These are the only two forms we have to consider. Which of these is the protonated form?
The top.
That's right, and this is the deprotonated. So this can't be right because this doesn't match the two forms. This is not one of our choices. So let's try again. What's the amine side going to look like at a pH of 11?
NH2.
Protonated or deprotonated?
Deprotonated.
So do you notice the difference between the carboxy side and the amine? When the carboxy is deprotonated, it's negative. But when the amine is deprotonated, it's neutral. So they're treated differently. Here's the when the amine is deprotonated, it's neutral because the protonated form was positive. So what's the charge on valine at a pH of 11?
Neutral.
Okay. No.
Negative. Negative.
Overall, it would be negative. Let's try drawing valine at a pH of 1.
pH of 1. At a pH of 1, then the oxygen would be in this protonated form. And
so what would be its charge?
Neutral. And the amine group would be and it's a protonated form.
How do I draw that?
H3 plus.
So what would be the charge on the whole valine?
+1.
Yeah. All right, so this is pretty sure to be on the exam. This is a popular type of problem here, changing the pH and drawing the amino acid at different pHs. So here we're using some of the acid-base material we were going over earlier. Okay, if they wanted to ask you a harder question, they could ask you what would be the ratio of the protonated to the deprotonated forms at different pHs. We did a little bit of an example of that. You would use the Henderson-Hasselbalch equation for that, but we haven't had time to look at that in more detail. But here's the basic type of question that you could see. Okay, so like I said, you should memorize these numbers. pKa of 9 for the amine and pKa of 2 for the carboxy side. Those are those ballpark numbers. And like the next step is to memorize all 20 of the amino acids. Now, when you're writing the amino acids, at first, you can write the whole picture. Once you get the hang of it, you can start saving time by only writing the side chain. So remember that all the amino acids have the same main chain. So once you understand how to draw the main chains, you can save time and just practice drawing the side chains. But you do have to memorize all 20 of those side chains and the 3-letter abbreviations. Okay, and that'll be important for us to work on that going forward. So what do you need to do on your own? You need to memorize the 20 amino acids. Next time when we meet again, we're going to be continuing to work on proteins and amino acids, and that session will be far more productive if we can practice the amino acids that you've memorized. So the next session will be far more productive if you have the amino acids memorized, and so we can practice and reinforce them during the session. And the last thing is, remember, you should also be making time to review your tutoring notes.